It's Debbie.
And Callie.
And we're on the Poquonnock River today in Groton, Connecticut. And we are going to be doing a little sampling for some bivalves. Before we get started though, I want to take a minute to ask people to share where they're watching from this morning. We want to particularly shout out our teachers this week. It is Teacher Appreciation Week, and there has never been a time where we appreciate teachers more than we do during this trying time. So we're so grateful if you're watching and sharing your materials with your students, and we're appreciative of all the hard work you're doing to make school a reality for them, even in a distance. So go ahead and share in the comments where you're watching from. And Callie is going to start to tell us how to use the piece of equipment that we're going to be sampling with today.
Yep, and actually before we do that, Debbie, I want to give a special shout out to some of the SMSP students, alum at least, that may be watching from home. I hope this looks very familiar to you, our sampling method today. I hope it reminds you of warm days back at Project O at summer camp. But the method that we're going to be using today is called tonging. And so we have it out of the water because it's actually quite a large piece of equipment. And it's basically two rakes attached to these long poles. So the idea with tonging is you can sample for shellfish in deep water. And so we're actually going to be deploying this sampling gear from the skiff, right from the bow deck here. So Debbie, if you want to show them the rake, I'm going to sort of move it back and forth. The idea is when it goes into the water, it's going to be in this open position. I'm going to push it down into the sediment, so we sample a big chunk of that sediment, and then I can close it back up again. And it's pretty heavy, but we'll pull this back onto the boat and then put it in the screens. We call this our muck board. Basically, it has some screens so we can filter away the sediment and see what kinds of organisms might be in there.
Oh, great. So what kind of bottom type is good for this?
This works really good with a muddy bottom. It can be used in sandy bottom too. It's a little less work because most of that sand filters away by the time you get it back onto the boat.
Great. So what kinds of things do you think we might find? Go ahead, share in the comments what sorts of things might be living in the muddy bottom of the Poquonnock River here. I talked a little bit about what our hope to find is, but we don't actually have a target species for this piece of equipment. We were hoping to find anything that's living at the bottom today, but we'll see what's down there. So share in the comments. I did get a comment already asking if the shellfishing is open to the public right now. I'm going to kind of turn around here and face the other side of the river. I'm not sure. Hi, Captain Ian. I'm not sure if you're able to see. I'm going to try to do a pinch zoom. Over in this area, there's a red sign that indicates that shellfishing is not open in this area right now. So we are using our scientific privilege, I suppose, to be able to sample here today. There's the pinch zoom. Okay. So again, shellfish are not open, so we're not going to consume any of the animals we catch today. We're going to release or use them for education purposes only.
All right, so we'll show you how this works. We'll see what we can find. I've got the tongs opened. I'm going to go to the edge of the boat, send them all the way down to the bottom.
Kind of murky here. I can't see the tongs already.
But I can feel them at the bottom. I can feel it kind of sticking in. So now I'm going to try to take a bite out of that sediment.
Okay, we got a couple predictions of clams or maybe crabs inside these tongs. We'll see what we find. All right. Oh, those look heavy.
Yeah.
Oh, nice one,
Can you get a view of that?
Oh, I can. Can you lift it without me?
Yep, I can do it. There we go. Right into
She's really strong, guys. You got this.
Woo! For the boat.
Nice. All right, so you can see how well that grabbed all of the muck that's down there at the bottom. There's all kinds of stuff in here. So I'm actually going to hand the camera off to Callie.
Okay.
And we're going to get all of that muck out of there using just the water that's right here as well, and a little help from a hand.
Oh, Molly's hoping for a cockle today, and Iris is wondering if there's going to be any worms in here.
Oh, I hope so. I love finding worms in our mud. All right, so right now I'm finding some shells. I'm not sure if we've got any clams in here yet. Oh, or anything alive, I mean. Woo!
I think I'll do the next one on the other side, right? This one's getting
Yeah, this one's pretty full.
full of sediment over here.
Yeah, I'm gonna have to, what you didn't see is beneath this mud in this tray is actually a sieve. So water should be able to pass through it if the mud isn't too thick. We may have actually put it up too thickly to allow it to all pass through. All right, I think I got most of that out of there. So let me do a quick hand rinse. And I'll take the camera again, and we will do another sample on the starboard side of the boat.
All right, so let's shift over. All right, so opening up the tongs, sending them down to the bottom, pushing it in, and then taking a bite right out of that sediment. Here we go.
All right, here it comes. See what we can find.
Okay, I can see some shells.
All right. Oh, there's some stuff in there. All right. Get a good shot of that and pass it off to you. Thank you.
Yep.
All right, see what we can find in here. Oh, baby.
I see some little bits of seaweed.
Now I'm wearing my mask today, but I could smell a little something in here. It's a little eggy. So to me that suggests that there might be some microbe action in this mud, sort of breaking down the decaying matter, anaerobic respiration in here.
Ooh.
Oh, interesting stuff. I just want to make sure I get all the living things out of here, even if I don't get all the mud. Oh, there's something here. I think it's a rock. All right. So I'll do one more hand rinse before I take that back from you.
Okay.
And I rinse the deck too.
Okay.
Very good. All right. So, we could take a look at some of the stuff that's in here. We definitely have some living organisms. I felt some.
All right, I'm gonna help to push some of this through.
Oh, there is a lot of mud. You got that right, Eileen. Now I'm seeing some organisms in here. If you see one that you think is alive, Callie, you can kind of pull out empty shells too.
Yep.
All right. Ooh, okay. So we have an organism here. Yes, we did get a rock for sure. But that right there that Callie's holding in her hand is an example of the hard-shell clam, sometimes called a quahog or a quahog. Mm-hmm. So that is a pretty popular shellfish in this area, or actually the world over. They're delicious.
And this one's not alive, but take a look at this one. This is just a very young hard-shell clam as well.
I'm trying to stay out of my own shadow here.
Yep,
Okay.
sorry, it's not alive. It's just half the shell, but evidence that we see, a flourishing population here. This is a young clam, and the ones that we showed you over here are much older because of their size. Whoops.
So we're out in nature. We're out in the river, Callie. Would we consider this a wild-caught animal, or is this part of a larger seeding project?
Well, it's kind of a tricky question. It's a little bit of both. This area is managed by the Groton Shellfish Commission, and so each town is responsible for shellfishing if they have such suitable habitats. And so you have to buy a permit if you're a recreational shell fisherman or clammer, and some of that money goes back to seeding. So they actually seed this area. Popular places where people look for these animals, they will actually dump them in here to help encourage that population to grow.
All right, so if you're watching and you don't know that clams grow from seeds, just kidding, right? So they're bivalves, so they will release eggs, and then those can actually be raised for a short time in captivity and then brought here when they reach a certain point in their life cycle. So that's what Callie means when she says seeding, right? So she's bringing, or the town will be bringing young clams or other bivalves to the area. Now I know I saw some shells in the other sieve that are not hard shell clams. So I want to make sure we get a chance to come over here and look at that. All right, this one is real murky muddy.
Okay.
So we're going to have Callie help me push the mud through this.
Do you want to continue filming or do you want me to?
Oh, well, if you've got your gloves on.
No, I can.
Here, there we go. All right.
Just doing the clam dance
I
here.
don't mind getting my hands all muddy. It's part of the experience. At least for me. All right, so I'm kind of running my hand along the bottom here to allow water to move through the sieve. Oh, I got another hard shell clam right here, big boy.
Oh, that's a good one.
Yeah, I'm gonna rinse that real good and kind of put that with our samples.
All right. Oh, and this is the fun part. It's kind of a little bit of a mystery. You can't exactly see what's in here, so.
Got another guy.
Nice. Ooh, here's the little one.
So how big do they need to be before recreational fishers are allowed to keep them, Callie?
So for the hard shell clam, it's usually you have a 1-inch ring. And if the clam does not slip through that 1-inch ring, then it's good to go. You're able to take it to eat.
So if it's too big for the ring, it's perfect for your plate.
Oh, this is a great sample.
Yeah.
Hard to see with another clam.
You couldn't
Hard
tell
shell.
before, right? But now there are lots of clams
There we go.
in here, yeah. So the shells that I was thinking that I saw before are a different kind of bivalve. Now this animal is not alive anymore. So these are just the empty shells. Does anybody at home know what kind of shells these are? They're a little murky, but this would have been home to different organisms.
Mm-hmm. Another bivalve, we'll give you that clue.
Yep, it's got 2 shells. That's what bivalve means, 2 shells, 2 halves to their body here. Mm-hmm. So this could have been one whole organism, the 2 shells of one. I don't know if we're getting any responses yet, but these are kind of polished smooth. You can see there's sort of a shiny interior to them. That's called mother of pearl or nacre. That might be a little hint to you if you know what kind of animal makes a pearl.
Well, Kyle and Marissa both got it. They're talking oysters. And that's right.
Yeah. And we're big fans of oysters here at Project O. We actually had a couple oysters at Project O that we brought with us today. So this is what they look like when they're alive. We didn't find any living ones in our samples, but it's not completely unreasonable that we would have found them there since we found the shells. Mm-hmm. And honestly, it's a great thing that the shells are here too, because they do kind of a reef building for the bottom substrate. So other animals can attach to these oyster shells or use them as habitat. So we're going to return most of these shells back to the water because if there are hiding places and attachment places on the bottom, that increases the biodiversity of the area here. So we're going to allow most of these shells to stay.
Oh, I'm seeing another one that's catching my eye. Yep, I think you've got half and I've got half here too.
Nice. What kind of clam is that?
Ooh, this is one of my favorites. Some of the viewers at home are getting a little hungry, I think, looking at all these bivalves. This is one of my favorites to eat. This is a soft shell clam, or a steamer is another name you may have heard. And so, again, they like that sediment around here as well. And this is another viable species in the area.
Right, so they also like a muddy bottom. So they kind of go into the mud a little bit different than our clams. And our oysters like to attach to other hard surfaces. So if we were to hope to have oysters in here at some time, we're going to put those oyster shells back in there. Oh, look at this. Right. Now I'm hoping to find a worm I think it was Iris was looking for a worm. I know I saw one earlier, but they're actually small enough to get through the holes in this sieve. So we might strike out. But there are lots of organisms that live in the mud here, and they're all doing important jobs for the community here. So our bivalves are all suspension feeders or filter feeders. So since they're living on the bottom, they can't do, here's a perfect example of an empty one, right?
A medder, that's what we call that.
They can't go out looking for food. You don't see a clam walking along the bottom looking for food. So what kinds of things do you think it could eat if it can't leave its spot in the mud? It can't go very far. It can go up and down in the mud a little bit, but
right. So the clam, they have a foot. Think of it kind of like a tongue, right? It's really muscular, and it helps them to dig and move around a bit.
What kinds of things? Go ahead, post in the comments if you think you know what a clam might be eating. If you're a filter feeder, what could you be filtering out of the water that's good to eat?
And while you're thinking about that, oh, Franklin is watching. Hi from
Franklin.
But Iris actually had a question. She said she thought pearls can grow in other bivalves. Is that true? And I can attest, I have found clam pearls before.
Ooh.
So in hard shell clams, those quahogs like we've been showing you, yeah, they can definitely grow pearls as well. So good question, Iris.
So the way that's formed is just if there's something annoying inside the bivalve shell, they can cover over it with that layer of nacre, and they can continue to cover it and cover it and cover it. Instead of having an annoying piece of sand inside their shell for their whole life, they can kind of cover it, and then it becomes a smooth pearl. So that process is famous for oysters, but you're absolutely right. Lots of things can do it.
Nice.
Do we have any guesses about filter feeding yet?
Yep, filter feeding, we have bacteria as a guess and also plankton.
Plankton, absolutely. They like to eat a lot of phytoplankton. So, ooh, I also found a piece of glass. Oh dear. That's a really important part of this environment. So if there's a lot of plankton, in fact, too much plankton, the bivalves can actually help improve the water quality of the environment. So how could there be too much plankton? What kinds of things would cause that? Since we are so close to land here, it's kind of easy to see that anything coming from the land might end up in the water, especially after a big rain. So things from the land, including like fertilizer or excess nutrients from the watershed, could end up in the river. And then if those nutrients are fertilizing all of the phytoplankton or the plant-like plankton, they're going to have a bloom. And if there's too much plankton, then all of a sudden the oxygen levels are disturbed in the river, and we could end up with dead zones. So by having a healthy bivalve population, we can help preserve the water quality and the water clarity, because I'll tell you what, the clams will take in some of that murky suspended sediments too, some of the mud that's carried through the river as well. So they're doing an important job. And then also the worms that we're hoping to find in here are doing a different important job. They're breaking down the organisms that are dead in the mud and kind of converting them into pieces that can be broken down by bacteria. So that stinky smell was from the bacteria or microbes breaking down the decomposing organisms. And then the clams need those nutrients because they feed the phytoplankton that they need. So it's kind of like all part of the same cycle here.
Yeah. I want to say good morning to Heather, who's watching, and also hi to Jill. Thanks for tuning in.
Got this one.
And we've got a couple of other things here that we want to
show that we get to see all these organisms.
Yeah. So far we've spotted hard-shell clams, right? The quahogs. We've spotted evidence of the soft-shell clams over here. We have seen the eastern oyster. Again, not a living one, but we pulled up a bunch of shells in our sample. There's one other bivalve that I want to, actually two more that I want to make sure that we highlight here as other species that we could potentially find here in the estuary. So right now, these are blue mussels that Debbie has in her hand. So again, bivalve, something that's definitely edible. And we didn't find any here. There's a few spots in the Poquonnock River that we've seen them before, but they usually like a rockier area or a sandier area as opposed to this really muddy spot that we're in right now.
I'll just show you how they attach to those rocks. Most of these have come loose, but they form these really strong threads called byssal threads that they use to attach to rocks and other things, or each other. So muddy spots, not so great for them, but rocky, definitely.
Yeah. And I think we had a question about scallops, too. So take a look at these beauties.
These are all bay scallops.
Yep, so that's the species that we find around here. Let's just shift them to the sunshine, Debbie. That would be great. I think
I know, I'm
easier to see.
I'm going to step out of the shot here. So. My shadow.
Okay, so we often find these shells on the beach. Once in a while, we find living ones here in the Poquonnock River. But the tricky part with tonging for scallops is they can swim. So they can actually kind of flap their shells or move their shells back and forth. There we go. And they can swim.
Just like they do in SpongeBob, seriously. They do swim.
Yep. So that tonging is usually not the best method for collecting those bay scallops.
So some other ways of collecting bivalves. Tonging is kind of the old-fashioned way, although now they have something called patent tonging, where you can deploy it from a boat and a big machine will do the tonging for you. So that's a nice convenience. There's also dredging, which is where you drag something behind your boat and get a real big glump of mud that way too. So those are typical ways of collecting bivalves in the modern day, although we're big fans of the tongs.
Or you can go old school, and I learned to go clamming just with my feet. So walking around in shallow water at low tide, finding them with your feet, or probably you've seen people using a clam rake, right, which is sort of a smaller,
kind of like this, but you can
smaller version,
broom-sized.
Yeah.
This is like the giant set, so we can deploy it from a boat and get into deeper water. All right, so a couple more organisms that we did not find around here, although they are native. We just didn't find them today. We have a nice collection here of a couple more types of bivalves that are popular. Now, these are another one that are edible. These are called razor clams, and they're long, and so they'll actually burrow down into the mud too. We got another example of those. Lots of little teeny mussels. These ones are adorable. These are also blue mussels. We also have some mussels around here called ribbed mussels that we'll find sometimes in the marsh. So maybe we'll do a marsh study soon. But these are the ones you're most likely to eat if you eat local mussels. And we said these ones are sometimes called angel wings. And you can kind of see why. They look like they could be attached right to the back of an angel, another type of clam. And then we have, these are called transverse arc clams. And they don't really get a whole lot bigger than this, maybe about that big, full grown. So there's not a lot of eating in them, but they are commonly found. We find them a lot of time in our Peterson grabs when we deploy them from the Enviro lab. So when we're collecting mud aboard our research vessel for looking for organisms, we call that the Peterson grab. It's a big orange claw. Maybe we'll do that sometime soon too. And so we'll see lots of cool organisms living in the mud all over the estuary. So when we're here in the estuary, we like to think about like the big picture. So we're thinking about runoff from the watershed, and then all the way down that way, the river will meet up with Long Island Sound. So one of the best parts of this part of the estuary is the fact that it's constantly changing, right? The depth is changing with the tides, the salinity, how salty it is, the temperature. It's always changing in here. And so there's a lot of different kinds of environments for a lot of different kinds of animals. So I think we're just about out of time, but please, if you have more questions, post them in the comments. Kyle and Marissa are going to be seining for minnows on Thursday in the salt pond, so definitely tune in for that. And that's 9:30 on Thursday morning, so we'll see you then.
Bye.
Bye.